Okay, let's dive right into this explainer. We're going to explore the absolutely awe-inspiring microscopic chemical factory operating inside our brains, and more importantly, how exactly drugs and toxins manage to completely sabotage it. Think about it. Literally every single thought you have, every movement you make, and every memory you store relies on this one fundamental biological process called synaptic transmission. Today, we're popping the hood to see how the whole shebang actually works. To teach this in depth, here's our roadmap for today. We'll start inside the synaptic machinery, then look at making and releasing messengers, followed by receptors and the cleanup. After that, we'll meet the hackers, agonists and antagonists, see what happens when drugs hijack the synaptic cleanup, and finally look at disrupting storage and release. Section 1. Inside the synaptic machinery. To get a chemical message across that tiny gap between neurons, what we call the synaptic cleft, 4 highly coordinated steps have to happen in perfect sequence. First, the sending side, the presynaptic axon terminal, has to synthesize and store the neurotransmitter. Then it has to actually release it. On the flip side, the postsynaptic target has to bind that message using its receptors. And finally, the system has to clean up the mess so it's ready to fire all over again. Section 2. Making and releasing messengers. Right off the bat, we see a huge difference in how these chemical messengers are actually manufactured. Small molecules like your amino acids and amines, think dopamine or serotonin, are built right on site. The enzymes needed to make them are shipped down to the axon terminal, where the transmitter is locally synthesized and packed into these tiny bubbles called synaptic vesicles. But peptides, they're much larger molecules. They actually have to be manufactured all the way back in the cell body's rough endoplasmic reticulum, processed in the Golgi apparatus, packaged into large secretory granules, and then physically hauled a massive distance all the way down the axon. Okay, so once these chemical messengers are packaged and waiting at the terminal, they need a signal to deploy. When an electrical signal, an action potential, comes zipping down the axon, it pops open voltage-gated channels. This triggers a highly precise biological mechanism known as exocytosis. The vesicle membrane fuses with a presynaptic membrane at a specific spot called the active zone, literally spilling its chemical payload right out into the synaptic cleft. What's really fascinating here is exactly how that trigger mechanism works. Under normal conditions, calcium levels inside the cell are kept incredibly low. But those voltage-gated channels I just mentioned, well, they're calcium channels. So when the action potential hits, a sudden, massive flood of calcium rushes in. There's a specific protein sitting on the vesicles called synaptotegmin that acts as a calcium sensor. The second it detects that flood, it instantly commands another set of proteins, the snare proteins, to tightly dock the vesicle and force it to release its contents. It's a microscopic marvel of engineering, honestly. Section 3. Receptors and the cleanup. So the neurotransmitters are released, they drift across the cleft, and they bind to receptors on the receiving cell. You can think of these receptors as 2 fundamentally different types of locks. On one hand, we have ionotropic receptors. These are transmitter-gated ion channels. The chemical key goes in, and boom, the door immediately pops open, allowing ions to rush in for a super fast 10 to 100 millisecond response. On the other hand, we have metabotropic receptors. When a transmitter binds here, it doesn't just open a door directly. Instead, it activates G proteins inside the cell, which then kick off a slower, much longer-lasting biochemical cascade using second messengers. Now, for those fast ionotropic channels, the type of ion that rushes in dictates everything. If the channel lets sodium flow into the cell, it causes depolarization, what we call an excitatory postsynaptic potential, or EPSP. Basically, a go signal. But if the channel lets negatively charged chloride ions rush in, it causes hyperpolarization, an IPSP. This is the ultimate stop signal, making it much harder for the receiving neuron to actually fire. But you can't just leave those transmitters sitting in the locks indefinitely. The factory floor has to be cleaned immediately. If transmitters linger, they cause constant activation, and eventually, the receptors just desensitize, and the receiving cell stops listening altogether. So the brain clears the cleft in 3 ways. First is reuptake, where specialized transporters vacuum the chemicals right back into the presynaptic terminal. Second is enzymatic degradation, where enzymes physically shred the transmitters apart. And third is just simple diffusion, where the molecules drift away on their own. Section 4. Hackers, agonists, and antagonists. Now, the crucial point is that a lot of drugs act directly on these receptor locks. They generally fall into 2 categories. Agonists are your lockpicks. They structurally mimic the body's natural neurotransmitters well enough to bind to the receptor and activate it. Nicotine does this to acetylcholine receptors. Opiates like morphine do it to opioid receptors. Antagonists, though, are like broken keys jammed into a keyhole. They bind to the receptor but don't activate it, effectively blocking the natural transmitters from doing their job. A terrifying example is curare, a poison that blocks receptors in your muscle tissue, leading to total paralysis. No way to move, even if your brain is screaming at your muscles to work. Some drugs, however, are a bit more subtle. They don't act as the primary key. Instead, they bind to secondary sites on the receptor to tweak how the lock functions. Take the GABA-A receptor, which handles inhibitory stop signals in the brain. Benzodiazepines bind to it and make the channel open more frequently, which gives you that anti-anxiety effect. Barbiturates bind to a completely different site and make the channel stay open longer, producing deep sedation. Even ethanol, alcohol, enhances this receptor's function in specific brain regions, powerfully boosting those inhibitory signals and slowing everything right down. Section 5. Hijacking the synaptic cleanup. By breaking the cleanup crew, some drugs deliberately leave neurotransmitters in the cleft to wildly intensify their effects. Cocaine and amphetamines, for instance, block the reuptake transporters for dopamine and norepinephrine. The transmitters are released normally, but the vacuum is broken, so they just sit there, continually hammering the receptors and leading to intense stimulation. Antidepressants, like SSRIs, think Prozac, do the exact same thing, but they specifically target the reuptake of serotonin. Alternatively, you can sabotage the enzymes. MAO inhibitors destroy the enzymes that normally break down serotonin and dopamine, causing a massive accumulation. And this mechanism really highlights the deadly severity of industrial sabotage when it's applied to our muscular system. Nerve gases and certain insecticides irreversibly bind to and inhibit acetylcholine esterase. That's the enzyme responsible for cleaning up acetylcholine at the neuromuscular junction. Without that enzyme, acetylcholine absolutely floods the synapse. The muscle receptors become massively overstimulated, then violently desensitize, and eventually fail completely. The result isn't just a muscle twitch, it's total respiratory failure and ultimately death. Section 6. Disrupting storage and release. Finally, let's look at the hackers that stop the message before it can even be sent. These diverse compounds hack the system right at the source. Reserpine, for example, goes in and interferes with the actual loading of neurotransmitters into the vesicles. It's essentially sending empty cargo ships to the cell membrane. THC, the active compound in marijuana, does something totally unique. It acts as an exogenous retrograde messenger. It binds to presynaptic CB1 receptors and actually tells the sending neuron to suppress the release of its own transmitters. Kind of wild, right? And then there are botulinum toxins. Remember those snare proteins we talked about earlier? The ones that dock the vesicles to the membrane? Well, botulinum toxins are enzymes that literally digest and destroy those snare proteins. The vesicles are full, the calcium rushes in, but the docking machinery is just totally melted. The neurotransmitters can never leave the cell. And yeah, the very same Botox used cosmetically to smooth wrinkles works by fundamentally destroying the docking machinery of the synapse, chemically paralyzing the target muscles. Seeing these mechanics laid bare leaves you with a pretty profound realization. When we look at this intricate dance of synthesis, calcium triggers, receptor binding, and enzymatic clearance, we're looking at a system of unimaginable precision. But as we've seen with these chemical saboteurs, it really leaves you wondering if a single molecule plugging a microscopic receptor can completely alter our consciousness, our movement, or end life itself. Just how incredibly fragile and perfectly balanced is our mental and physical existence day in and day out.